Hello, everyone, and welcome to today's webinar, Visualizing Membrane Transformation in Live Cell with STED, Fusion and Budding. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit www.leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. Also, if you experience any technical questions or issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Dr. Ling-Gang Wu, Senior Investigator, Synaptic Transmission Section, NINDS, NIH. Dr. Wu, you may now begin your presentation.
Thank you very much for your introduction. Today, I'd like to share with you our journey towards visualizing the membrane transformation of fusion and budding. As you may know, fusion and budding is essential for every cell's life, like in the intracellular trafficking of the cells, in the regulation of the blood glucose levels that is relevant to diabetes, in viral infection, including the coronavirus infection, the COVID-19 pandemic, in the release of a transmitter that mediates the synaptic transmission essential for our brain's function, and in the fight or flight responses. This was a picture taken in the Yellowstone National Park. I was about four meters away from this guy, and my release of the adrenaline must be really high. So I was in the flight response instead of the fight response. The most used study, the most used preparation for the study of fusion and budding is the synapse, where you have about 200 vesicles or so in the nerve terminals. And these vesicles contain neurotransmitters that can be released out via exocytosis to mediate synaptic transmission. And after that, then the endocytosis will occur to recycle these exocytosis vesicles and therefore to sustain synaptic transmission. Back in 1970s and 80s, John Heuser and Tom Rees discovered these fusion pore-like structures, used the electron microscopy methods. They then proposed the full collapse fusion hypothesis, which involves the fusion pore opening and then fusion pore dilation until the vesicle is completely flattened at the plasma membrane. After this full collapse fusion, they saw this morphological change that reflects endocytosis. Therefore, they suggest that the endocytosis undergoes these morphological changes that is almost the reverse of this full collapse fusion. And it takes place at the endocytic zone, which is different from the exocytic zone here. At about the same time, Saccarelli and his colleagues proposed the kiss and run form of the fusion, which involves fusion pore opening and closing very rapidly, and the pore usually is very narrow so that it may limit transmitter release. And their hypothesis based on the observation of this omega-shaped membrane profile with the electron microscopy method. 2 decades later, in 1990s, the full collapse fusion followed by the slow endocytosis has been very well accepted, whereas the kiss and run form has been debated for a long time without being generally accepted. Both of these hypotheses have not been observed directly in live cells. So we set out to revisit this issue by asking whether we can see the membrane dynamics in live cells, and if so, their underlying mechanisms and functions. So my lab starts at about the beginning of 2000s. At the time, there's no super resolution microscopes, fast super resolution microscopes available. So we use the electrophysiological setups to study the membrane dynamics. We're using the capacitance measurements. The capacitance is proportional to the membrane area. When exocytosis occurs, the vesicle membrane is added to the surface area, to the surface membrane. Therefore, the surface area is increased. So an increase of exocytosis means that the capacitance increase, and the decay of the capacitance means endocytosis, because endocytosis retrieve vesicles from the plasma membrane. So to recall capacitance, we PET clamp the large nerve terminal, calyx of hair nerve terminal, this nerve terminal allows us also patch on the postsynapse cell, and this is in the auditory brainstem. So my lab's first project is to establish the capacitance measurement technique in the central synapse. And here we see the capacitance increase that is corresponding to the EPSCs recorded simultaneously at the same synapse. So the EPSC reflects the transmitter release that acts on the transmitter receptors. And therefore, this capacitance jump means the exocytosis. With this establishment of these techniques, then we record endocytosis. We see the endocytosis, slow ones and fast ones, which takes about seconds or so. So this has been interpreted as kiss and runs, but some people will argue that it could still be caused by the Fred to wrong transition, very fast Fred to wrong transition. At the time, we don't know which one is right. Now, this is a wholesale recording, so this jump here reflects tens of thousands of vesicle fusions and endocytosis, so ensemble average of from the whole nerve terminals. Here we try to record the single vesicle fusion and endocytosis by recording the minis and simultaneously the capacitance increase here. We could not see any capacitance increase because the single vesicle fusion will cause a very tiny capacitance increase. So to get to the single vesicles, we have to average about 320,000 minis to get a very tiny jump here, about 65 attofarads of the capacitance increase and decay that reflects endocytosis. So notice here that the scale here is about 10,000 times less than the scale here. So this is a single vesicle fusion. And this is really laborious because it takes our former postdoc, Xin Wu, about 1.5 years just to get to this trace. To see the single vesicle fusion with a single trace recordings, we perform this cell-attached recordings on the small vesicles with also the fusion port conductance measurements. And to do that, we have to remove the postsynapse cells using a large pipette to pull the postsynapse cells out, and that will expose the release phase of this calyx. Then we use another pipette to patch on the release phase of the calyx in green. And then we hope that the vesicle will fuse on the patch membrane. And under this condition, then we can determine whether it's kiss and run or full collapse fusion. Here, I'll show you an example of the kiss and runs, which involve the capacitance increase and decrease, and that is accompanied by the fusion port conductance increase and decay. So this provides a strong evidence for the kiss and run at synapse. At about the same time, we also see the fusion port conductance increase that expands to a certain value that we could not detect. And this was accompanied by the capacitance increase, which is not followed by the down step. So that is consistent with the full collapse fusion. And sometimes we also see the giant capacitance increase. Here, the scale bar here is 20 times than that. And this giant capacitance increase is because of the vesicle-vesicle fusion that forms the compound vesicles. And this compound vesicle then undergoes exocytosis to cause a gigantic increase in the capacitance. We can also see this compound vesicle formation in the electron microscope. This compound vesicle fusion causes very large miniature EPSCs and therefore increase the quantal size. So we spent about 10 years in trying to understand the fusion modes with the capacitance and conductance measurements. And I should say that these are most difficult experiments in the synaptic transmission studies, at least in my view. But this is still not the direct measurements. Our resolution is limited to about 5 nanometers of the fusion pores based on the indirect estimate from the capacitance and the conductance measurements. It is not direct, so anything above 5 nanometers is still not resolved, and therefore we can still put back these question marks here. So we have not been able to resolve these membrane profile changes. So by this time, 2010, the STED microscope is available. It has a resolution about 60 to 80 nanometers and every 26 to 100 milliseconds or so. Now this resolution is not sufficient to resolve the small 40 nanometer vesicle fusion and profile changes. So we have to use a different preparation, the bovine adrenochromin cell, this cell is a neuroendocrine cell. It's similar, has a similar exoendocytosis as neurons, but it has the very large vesicles, 300, 400, or 500 nanometer vesicles shown here. So we should be able to use the STED microscope to resolve this membrane profile change of these vesicles. We then culture the cells on the cover slips, and we add a dye like Alexa 647 or 532 in the bar solutions. And we see this dark area. This reflects the cell's footprint sitting on the cover slip. It's dark because you only have about a very thin layer of the cell membranes, the gaps between the cell membrane and the fluorescent solution, and then the cover slips. So very small gap here for the fluorescent molecule to be here. Therefore, you see it very dark. And when vesicle fusion occurs, then this fluorescent dye can then diffuse into the fusion vesicle, so we can see a spot coming after the fusion. To induce fusion, we PET scan the cell, we give a depolarization, 1 second depolarization, and that causes calcium currents and capacitance increase that reflects exocytosis and decay that reflects endocytosis. So I'm going to play a video of this process. So at the beginning, there will be nothing here. It's a resting condition. Then we give a depolarization. You will see the spots come in because of the fusion. So right now, start. Now you see the spots come in. That's because of the depolarization that evoke these fusions. To see vesicle fusions, membrane profile changes, we overexpress phospholipase C delta 1 PS domain attached with NBN green or EGFP. And this probe binds to the PIP2 at the inner leaflet of the plasma membrane. So with the STED microscope to scan on the XZ plane, which means it's perpendicular, it's proportional to the screen of this computer, we will see the stretch of membranes that is sitting on top of the cover slips, as just like the drawing here. If we label the vesicles with a fluorescent transmitter, then we will see the vesicles docking on this green membrane. And when vesicle fusion occurs, then this green probe will then diffuse into the fusion vesicles and therefore to light up the fusion vesicles membrane profile. So in the experiment, we also add to 532 or Alexa 647 in the bar solutions. And we then do the axis plane scanning. So now you see a stretch of the green membrane sitting on top of the solutions, ATO 532s. And then underneath the ATO 532 is the cover slip that you don't see. After the fusion, then we will see that the red dye will diffuse in, and the green dye will also diffuse into the vesicle to label the vesicles. So we give a depolarization, we see the fusion events, and this is what we see here, the membrane profiles together with the spot of the red dye because of the diffusion of the red dye into the fusion vesicle. And here I'll show you a video, a real-time video of this process. So this means green omega profiles and a spot that fills the cavity of the fusion vesicle. Sometimes we'll see the green vesicles come in first, followed by the red spots appear, as shown here in this real-time video. Green spots, followed by the red spot. And that's because of the hemifusion that allows for only the green dye to probe the membrane to diffuse into the hemifused vesicles, but not the red dye to diffuse through the fusion pore. And when the fusion pore opens, then the red dye gets in. So there's a delay here, you see. It's hemifusion to full fusion. Sometimes the hemifusion does not proceed further into full fusion. So you see only the green dye appear here, green probes here. So this is showing you the green signals, but not the red signal. That's because of hemifusion that does not proceed further into full fusion. So we have established this hemifusion pathway in live cells. After the hemi to full fusion, we see sometimes the fusion pore expansion as shown here in this example. And this shows you that the fusion pore expansion could be really fast and be below beyond our resolutions, 0.1 seconds, 46 milliseconds, or even 26 milliseconds, we see the rupture of the fusion pore here. The fusion pore shape will be oval or round shape. If we cross the fusion pores perpendicular to the screen here, we'll see this oval shape of the fusion pore. After the fusion pore expansion, the fusion pore can then close very rapidly, as shown here in this example and in this video. Here, fusion pore opens and closes very rapidly, and this will be repeated again. Fusion pore open and close. The opening up to about 200 nanometers and then close. So this is really surprised, because what we understand before is that fusion pore has to be very nano in order for them to be closing again. If it's beyond like 10 nanometers, most people will think that it will not be able to close. Now the closure is accompanied by the breaching of the red dye, as you can see here, and that's because of we gave a very strong excitation to the red dye, and when there's a limited number of the red dye in the closed vesicles, it can be all breached out. So you can this breaching of red dye confirms that it also means the fusion pore closure. And when the dye is not breaching, it means that the fusion pore is not closed, shown here in this example. So sometimes the fusion pore will stay there for a long time without being closed. This is another example. So no closures in this case. Next, we'd like to know whether the fusion pore will undergo the full collapse fusion by dilation of the fusion pore or by something else. And what we found is, surprisingly, that it is not by the full collapse fusion as generally believed. Rather, it is undergoing this string fusion as shown here in this example here. And you can see it better in this video, real-time video. You see the fusion pore, not dilations, rather the omega profile is shrinking. So we call this string fusions. And all of these videos are shown after the depolarization. So they're all evoked by the depolarization. And this I'll show you another example, because this is so striking. So I'm going to show you another example of the string fusions. Okay. So sometimes the vesicle decided to enlarge instead of the shrinking, as shown here in this example. And here I'll show you a real-time video of this enlarged fusion. So vesicle fusion, then it slowly enlarge instead of shrinking. Repeat it. Sometimes we see the vesicle fusion on the recently fused vesicle, and this is called the sequential compound fusion. Here I'll show you an example of this process. The vesicle fusion occurs first here, and on top of these vesicles, there will be another vesicle fusion at some time later after the depolarization. Here is the real-time video of this process, sequential compound fusion. Fusion here. And that's another vesicle fusion, sequential compound fusion. So I have shown you different modes of fusion. Next, I'd like to show you the endocytic membrane transformations. This shows you the first observation of the thread to run endocytic membrane transformations. And here is the first video, real-time video of this process. So here's the thread membranes, and after depolarization, it will then be converted into an oval shape of the vesicles. First is lambda shape formation. You could see some spike as if something is pulling into the cytosol. Then some force is squeezing on the side. Now it becomes an oval shaped vesicle. The red dye is breaching, confirming that it's the fission occurs. We found that this thread to run transition is composed of three steps, three modular steps. One is the thread to lambda, lambda to omega, and the third one is the omega to O transitions. This example shows you the thread to lambda transitions, shown here in this real-time videos. You notice that there was some spike pulling, so there's a force that are pulling membrane inward, and you can see the growing of the lambda shape. This lambda profile does not proceed further into omega profile. It stops here. And because of this reason, we see these preformed lambda shapes and omega shape profiles in the resting conditions, as shown here in this STED imagings, also in the electron microscopic images. And these preformed structures, lambdas and omegas, can also proceed further to become an omega or the vesicle-like structures. So here show you an example of the preformed lambda shape that is converted into the omega shape after the depolarization, shown here in this real-time video. So that's lambda becoming greater in omega. There's a force that are squeezing on the side here. You can see the pore, omega's pore is slowly getting narrow. And this one does not proceed further to become a vesicle. And some of these preformed omega profiles can then proceed into becoming a vesicle by closing their pore after the depolarization, shown here as the gray triangle. We can see this better in this real-time video. So it'll be really fast. This pore, large pore is going to be closed after depolarization, and the red dye will be then bleached out to confirm that it's the pore closure. Show you here. It's open, close, and then red dye bleach. So I have shown you the three modular transitions that mediate the thread to wrong transition. They have to undergo this lambda profile formations and omega profile formations. And if the lambda profile is larger than the vesicle is larger. The lambda profile is smaller, and the vesicle is smaller. All right, so I have shown you the membrane fusion and budding in live cells that we can observe with the STED microscope. And also I show you that the compound vesicle fusions at synapse. Next, we would like to study their mechanisms. The first one is the endocytic membrane transformation. The thread to the lambda shape transition is mediated by actin and dynamin, because if we block them with the trunculin A cytokeratin D, dinosaur or dynamin K44A, dominant negative, we see the block of the thread to the lambda transition dramatically. And if we label the actin with live actin in red, then we see this F-actin, the filamentous actin, that appears to attach to the tip of the lambda shape and pull the membrane inward. You can see some of the examples here that the actin filament is on the tip of this growing lambda shape. And here I'm going to show you a real-time video of this process. Notice here, it is this very tiny spot here at the center of this lambda shape membrane, and this red dot is going to grow and become a filament. It looks like it's going to be brighter right now. Now it becomes a filament that appears to pull the membrane inward. Now, if we label dynamin in red, we see dynamin surrounds the base of the lambda shape. So this is the XZ-plane images of the dynamin in red that surround the side of the lambdas. And this is the XY images of dynamin in red that surrounds the green base of the lambda shape here. So here I show you an example that when lambda is growing, we see the dynamin surround in the base of the lambda and also recruited to the tip of the lambda shape, as shown here. So this is a cartoon showing you our model that the actin and dynamin can pull membrane inward by attaching at the tip of the lambda shape, and that will pull membrane inward. Our next question is how this lambda shape is then converted into the omega shape. We notice that the dynamin in red surrounds the base of the lambda shape. Also, dynamin in red surrounds the base and pore region of this omega shape, as shown here in this XY plane images across the pore region. We see dynamin in red that surrounds the pore region of the omega profiles, and this can be better observed here in this dynamic changes from the lambda to omega profiles. The dynamin in red is surrounding the base and appear to constrict together with the omega profile to reduce the pore size of this omega profile. So we can see this better in this real-time videos. Dynamic is in red and membrane profile is in green. So here, notice here the dynamic punctures here is surrounding the base and pore right now of the omega profiles. So dynamic appears to constrict the pore or the base of the lambda share and omega profiles to mediate the constrictions. And if we block dynamic with these two approaches, we see the block of the lambda to omega transition. All right. So this means that the dynamic can constrict very large lambda share. It could be like 200 to 800 nanometers. And what we know about dynamic is that it can constrict about 10 nanometer pores instead of these very large structures. So to show that dynamic can do so, we collaborate with Nidhi Kundu and Jenny Hinsiao. They show that dynamic alone can constrict the liposomes in vitro from hundreds of nanometers to tens of nanometers, shown here in these statistics. They also found that dynamic forms this helix to surround these large liposome-like structures. And so we propose that dynamic can then forms helix to constrict the lambda shape and therefore to mediate the lambda to omega transformation. The next dynamic then can continue to constrict the omega shape profiles pore to force it to become a vesicles. And this I'll show you the dynamics in red that surrounds the pore region of the omega profiles, and these two puncta will come together as if dynamic continue to constrict and force this omega profile to become a vesicle. We can see this better in this real-time video. Dynamic in red here is surrounding the pore region, and then after the depolarization, these two puncta will come together with the pore closure. You can see now it comes together. Dynamic is constricting the pore, close the pore. And if we block dynamic with dinosaur or K44A overexpression, we see the reduction of omega to O transitions dramatically. Therefore, we conclude that dynamic and actin are involved in pulling the membrane, and dynamic is involved in constricting the base of the lambda and omega profiles pore. So there are two forces here. One is the pulling at the tip, the other is the constriction. And our collaborator, Ben Zucker and Misha Kozlov, did the mathematical modeling to show that indeed two forces, the pulling force and the base constriction, is sufficient to generate omega shape profiles. So they show us a simulation showing that the pulling can then generate a lambda profile, and the constriction will then create a omega profile, as shown here. So here is the lambda shape formation by pulling, and the constriction will then change this into a omega shape. So with these results, we conclude that actin dynamic are involved in converting the flat membrane into lambda shape, then omega shape, and then the vesicles. I have shown you that the preformed omega profile can be converted into round vesicles after the depolarization. And this provides us the opportunity to screen the proteins involved in the fission process. Now here I show you more of these preformed omega profiles that are existing in the resting condition. They are waiting for the signals depolarization to be close their pores. So we screen the proteins involved in this fission process, and what we found is really surprised that we found clathrin is involved in the fission process. Clathrin has been well known to coat the vesicles, just like in this case is the coating of the vesicles bodies. These vesicles are very small, about 100 or nanometer or less. It is not known to mediate fission at all. When we inhibit clathrin by the siRNA or ssRNA knockdowns, we see the pre-close, this preformed omega profile closure decrease dramatically. It can also be decreased by a pit stop which blocks the clathrin polymerization. This means the clathrin is involved in the fission process. Next, we'd like to know whether clathrin is involved in the fusion pore closure. And we found indeed with these three approaches, we found that the fusion pore closure is decreased dramatically as well. So clathrin is involved in closing the pore. And because of this closure force, it antagonizes the fusion pore extensions. Therefore, it should limit transmitter release. And indeed, when we label the fluorescent transmitters in the vesicles and then look at their release rate, we see that this FFN release, we can see this FFN release, and this release rate is speeding up by the knockdown of clathrin. So here's a control dotted line. And after the knockdown of clathrin, we see a speeding up of the release process. That's because of clathrin's effect on closing the pore. So we conclude that clathrin is involved in closing the pore. Now the question is how clathrin closes the pore. Well, first of all, we label clathrin. We found that clathrin in magenta is localized in the pore region here of this omega profile. And here show you a 3D scanning of the clathrin in magenta. You can see clathrin surrounds the pore regions of this omega profiles. And when we cut through the pore region in the XY plane, perpendicular to the screen, we see this magenta clathrin surrounds the green pore regions here. And here show you more examples of the green pores surrounded by magenta clathrin. Some of these pores are very small that we cannot resolve with a STED microscope, but we can still see the clathrin surrounds these pores. Now, if we use the MINFLUX imaging to resolve single clathrin molecules, we can see the single clathrin molecule clustered together around the pore region. And this appears to be polymerized. Some of them are very close to each other, these single clathrin molecules. In collaboration with Kazuki Obasi, Min-sun, and Justin Taroska, we see these clathrin patches that is surrounding this omega profile, the membrane imagination. Some of them appears to be on the pore region. As you can see here, the clathrin polymerized around the pore region, just like what we see here as these omega profiles surrounded at the pore region by these clathrin patches here. If we use a clathrin mutation to block the clathrin polymerization, we see that the clathrin polymerization is reduced dramatically. Here we don't see any clathrin patches in these examples. And here is a statistics showing you that this clathrin mutant can block the clathrin polymerizations. This mutant also blocks the preformed omega profile closures and also the fusion pore closure. So this means that the clathrin polymerization is required to mediate the pore closure. Our collaborator Ehud Haimov and Misha Kozlov did the computer simulation and mathematical modeling to show that the polymerization of clathrin at the base and the pore region of the omega profile is sufficient to constrict the pore and to mediate the fission process. So it can constrict until the pore is very, very narrow. All right, so with these results, we like to conclude that clathrin is a new class of the fission enzyme involved in this preformed omega profile closure and fusion pore closure. Speaking of the fusion, one would like to know the fusion pore dynamics mechanisms. And by doing many experiments that I don't have time to talk about, we found that the fusion pore is controlled by two forces. One is the fusion pore expansion force, controlled by the snare proteins, actins, and tensions. The other one is the constriction force. It's controlled by dynamin and clathrin and calcium. And these two forces will compete with each other to decide the fusion dynamics, like if the fusion will stop at the hemifusion or proceed to become a fusion pore opening with a nanopore or with the expansion of the pore or fusion pore closure. Next, we'd like to know the shrink mechanisms. Why the vesicle choose to shrink instead of the full collapse fusion? Now, collaborator Satis Theo Karen, Risu, and Ben Oceanosi did a computer simulation. They found that the driving force is caused by this delta P, the intracellular to the extracellular osmotic pressure differences. There's a positive difference of this delta P, and this positive driving force then can squeeze the fused vesicle membrane to make the vesicle membrane crumple and lose the membrane tension. The higher membrane tension at the plasma membrane can then reel off the low tension vesicle membrane. So the vesicle becomes shrink, and is repeated. So you can see the vesicle shrink instead of the collapse. And this model will predict that if you reduce the driving force, the delta P, by applying a hypertonic solution, then the string fusion is going to decrease. And indeed, we see that experimentally the decrease of the string fusion by applying a hypertonic solution. If we apply latroncurin A to reduce the filamentous actin, and that because the filamentous actin is essential for maintaining the higher membrane tension at the plasma membrane. So this latroncurin A reduce the membrane tension. It will also reduce the string fusions, and we see indeed it reduced the string fusion because of the driving force for this higher membrane tension is reduced. Now, if we increase the delta P, the driving force, so this should rescue the inhibition by latroncurin, and indeed we see this rescue of the string fusions by the latroncurin. So we apply a hypotonic solution in the presence of latroncurin, and we see a much larger string fusions. Yeah. So with these results, we conclude that osmolarity, tension, and actin are involved in mediating the string fusions and to produce the string fusion instead of the full collapse fusion. All right, so I have shown you our large cell transitions and the mechanisms, molecular mechanics involved in mediating some of these transitions. Next, I would like to spend a few minutes to talk about the functions that we have not been able to recognize previously because of this complexity of these membrane transformations. The first one is the hemifusion, which can control the release probability, because if the hemifusion structure does not proceed further into becoming a full fusion event, then it causes the failure of the release, and therefore this can control the release probability. A delay in the hemifusion to full fusion events can then generate a synchronized release. Another way to generate a synchronized release is this sequential compound fusion. So here I show you an example that we loaded the vesicles with the fluorescent phosphorescent neurotransmitter FFN511. So you see two vesicles here, and after the depolarization, we can then see this vesicle will be diffusing out this release of FFNs, first one and second one, and here there will be the H-shaped vesicle formation. So I'm going to play this video. Fusions occurs, and then you see the second one, the formation of the H-shaped vesicles. Now it's repeated again so you can see them. So that's the sequential compound fusion that can cause a synchronized release. And of course, if the fusion pore is narrow, then it will control the release or limit transmitter release. So here we show you that the phosphorescent neurotransmitter release low if we cannot resolve the fusion pore. And if we can resolve the fusion pore, then the FFN release will be much faster than the one shown here in the left. Our study also leads to redefining the kiss and run. The kiss and run has been traditionally defined as the fast vesicle fusion pore opening with a narrow pore and then close very rapidly. Now it can be redefined as either the fast or slow fusion pore closure. The pore does not necessarily to be the very narrow one. It could also be very large to facilitate transmitter release, and the resulting vesicle could be either large or small. So these in large vesicle fusion can close their pore to form large vesicles. The the string fusion can be stopped here and close the port, partially shrink, and then close the port to generate smaller vesicles. So the KISN1 can control the vesicle size. Our finding of the string fusion challenges the full collapse fusion as the major mode of the exocytosis. And finally, I'd like to talk about the endocytosis. The endocytosis, when I show you these traces, most people will interpret this as the very fast Fred to long transitions. And here I show you more of the examples of wholesale recorded capacitance, no endocytosis, slow ones, fast endocytosis, ultra fast one that takes 1 second or less, and then the overshoots of the endocytosis. And most people will interpret these different forms as the Fred to long transitions, just like you see here, the Fred to long transitions. And what we found is really surprising here, that the Fred to long transition is minimal. Rather, it is the fusion pore closure and the preformed omega to O transitions. These two forms are the majorities. And here I show you a bar graph of some of comparison of preformed omega to O transition, which is much higher than the Fred to O transitions. And the same for the fusion pore closure. So because of this unexpected and surprising finding, we want to verify this again by reconstructing the population exoendocytosis from the individual exoendocytosis. So here is a single cells capacitance recording from single cells, showing you different forms of endocytosis, again, slow, fast, ultra fast, and overshoots. And at the same cell, we then recorded individual vesicle fusion and also the preformed omega pore closures. So if we see a fusion event, we give an up step, and if we see a fusion pore closure, we give a down step. If we see only the fusion pore opening without the closures, we give an up step here. And then we sum all these events together, we see the increase here that mimics the capacitance increase, and there's some decay here that may reflect the fusion pore closure of the endocytosis. We also recorded the preformed omega pore closure by giving a down step here, and we sum all them together in each of these cells. These are the number of events that we recorded from cell bottoms. So we see the decay here, and that's because of the preformed closure in each cells here. And then we sum these two reconstructed traces together, we get this red trace that is really closely resemble the wholesale recorded capacitance trace at the same cell. So this means that the fusion pore closure, together with preformed omega profile closure, mediates these different forms of endocytosis, like slows, fast, ultra fast, overshoots, or even no endocytosis. So again, it is not the Fred to long transition that mediates endocytosis primarily. Primarily, it is caused by the fusion pore closure as well as the preformed omega profile closure. So in conclusion, I have shown you the fusion and buttons membrane transformations, mechanisms, and functions. And this is rather different from the classical model that we started with about 20 years ago. And finally, I'd like to thank many, many people involved in this work. The electrophysiology is performed mostly by these people here, and Jian Sun and Li Minghe and Xin Zhe, who started the wholesale and the cell-attached recording of the capacitance at the calyx height. The imaging was performed by many people, in particular Wang Zuxin, Danny Hamid. Wang Zuxin is here, Danny Hamid here, Xie Chenjiang, and Wei Dong Zhao here, Peter Wen, and Jian Vito is now here, Xiao Li Guo, Li Siwei, Li Hao, Keith Chiang, etc. Most of the video are provided by Wang Zuxin. And here are my current lab members. I also like to thank many of my collaborators that I mentioned in my talk. And thank you very much for your attention.
Thank you, Dr. Wu, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions submitted today during the live and those during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care everyone. Goodbye.